Hello, my dear students. I hope you all have been doing well. Today we are going to discuss about experiment number 8b, and yesterday we have already discussed about experiment number 8a. In that we discussed about the quantitative analysis of one of the protein, that was gelatin, and here we would be studying about the qualitative analysis of one of the serum protein, that is the albumin. So before starting the discussion that how we can be perform an experiment by which we can identify the presence of albumin in unknown sample that would be provided during the experiment. So before discussing the experiment or actual test that would be necessary for qualitative analysis of albumin, first we have to understand that what actually is albumin, what albumin, what is actually this albumin, and where it is exist and what is its significance or importance. So before to understand the albumin, we have to first understand that we have to understand that albumin is a protein, and it is a plasma protein, and to understand about the significance of protein, we have known this fact that albumin is present in our blood, and one of the particular part of the blood, that is the plasma. So this is one thing, but before here we will be studying about what is the significance of albumin and why we wish to qualitatively analyze this particular type of protein in the provided sample. So we have to understand that where this albumin is found. Actually, the albumin found in the plasma. And what is the plasma? Actually, if we will go about the this picture, so this picture is explaining you that a human being, the total body weight of a human being is based on or composed on two parts. One is the blood, that is 8% of the total body weight, or the other fluids, tissue or other fluid encompassing 92%. And then here when we centrifuge blood, the whole from our body, total body weight, 8% of our body weight is blood. So a normal human being is having 8% of blood of its whole total body weight, overall total body weight. So if we centrifuge this 8% blood, so what we will be having, we will be, after centrifugation, we will be having two compartment, or the blood that we will be having in a test tube. After centrifugation, this blood would be separated into two layers, and one layer would be containing one layer that will the one layer that will not having you can say that blood cell. So that this will be the plasma, and the other layer and the plasma would be 55%. However, the other layer that will sediment at the bottom of the test tube would be consisting on the blood cells, and it would be 45%. And then what this and why this layer is we found why the layer that would be consisting on different body blood cell would be found at the bottom of the test tube, because this layer would be containing different blood cells that will be heavy in nature. So they after centrifugation they will sediment at the bottom of the test tube. So after centrifugation of the blood, we would be having two layers. One layer would be heavier, and it would be containing different blood cells, and it would be 45% of total blood. And however, the rest part, that would be 55% of whole blood, would be plasma, and this plasma would be containing 7% protein and 92% water and 1% of other solutes. So what solutes that can be present in this plasma? It would be iron, nutrient, means iron, and other nutrients. They may be waste products, some gases, or some regulatory substances. However, the 7% of the protein of this plasma would be different type of protein. In that 57 to 60% would be albumin, and other would be 38% globulin, and then fibrinogen would be 44%, and prothrombin would be 1%. So from here we can see that the albumin is means one of the greater fraction of the protein that would be present in the plasma. So the other second largest portion of the protein that would be present in the plasma, it would be globulin. However, this fibrinogen and prothrombin, these will be in the less less amount. So from here we can understand that how much albumin is important for our blood. And then the what type of blood cell can be found in the rest portion that would be found in the bottom layer of this test tube. These blood cell include the erythrocyte, leukocyte. Leukocyte are the white blood cell, and the platelets are the cell that involve in the clotting of blood. And this platelet would be 140,000 to 340,000, means one of the largest portion of the blood are the platelets. So it is another important part of, or another important component of the blood. However, the second important component of our blood is the leukocyte, that would be from 5,000 to 10,000, and these leukocyte are the white blood cell. And white blood cell means what class of white blood cell can be found in the blood. All these type they may be basophils, eosinophils, neutrophils. These are granulocyte, and then because they eosinophil, basophil, and neutrophil, these cells are called as granulocyte because they are having granules. So whenever they encounter any foreign particle, they release their granule, and they kill the foreign cell. And monocyte and lymphocyte can also be found in the blood cell. So all these leukocyte are make the immune system of our body. So whenever any foreign particle enter in our body, so first type of white blood cell that start moving towards the foreign organism would be neutrophils. So one of the major portion of the leukocyte that is present in our blood is the neutrophil, that would be present into 40 to 60%. So this is most important type of white blood cell that is present in our blood, and it is one of the most important component of immune system. However, the erythrocyte are present the red blood cell, and then these red blood cell may be found in a uh, in a human being, ranges from 4.2 to 6.2 million. So this was a whole million per cubic millimeter. So this was whole about the composition of blood. So from here we can this I showed you this picture just to give you an idea that where this albumin exists in our body. So now excuse me, so now we would be going towards our experiment. So now before going to discuss about the experiment, we also discuss a little bit about the significance of this albumin. This albumin is also important because it regulates our osmotic pressure, it neutralize means free radical, and it is also bind and stabilize a range of important small molecule and ions. And albumin actually one of the most important function of albumin is that it bind with the fatty acid, free fatty acid, and circulate it. So the circulation of free fatty acid is make possible due to this albumin. So this was some of the importance of albumin. And now we would be going to now we would be going to discuss about how we can analyze the component of albumin in provided sample. So how we can qualitatively analyze? So our objective is to analyze or is to identify the albumin in the given sample. So for this purpose we would be utilizing the few tests. So as we have already discussed that first to find out the to identify any protein in the mixture, first we have to analyze any particular type of protein in the mixture, we have to first analyze the presence of a protein. So if we would be able to find out the protein is present in the sample, but we did not about its means class. So it would be helpful for us first to find out the presence of protein. So how we can find out the presence of protein in the given sample to perform the test? That would give us the surety that in the mixture that is provided to us is containing the peptide bond. The first test would be to find out the presence of a peptide bond. So if the peptide bond are present in the sample, so it means there would be amino acid present, because amino acid peptide bond are found to join with the two amino acid together. So first test we would be perform would be Barrett test, and then the other test that we perform for the identification of amino acid would be the Ninhydrin test, and after that we would be perform the test to find out the presence of peptone in the sample. So basic question is here, why we are performing the test to find out the peptone? Because peptone is one of the first type of protein that found in any sample after digestion. So for then we perform these two tests, as Barge test and this test as Barge test for the presence of so we perform these two tests for the confirmation of protein in the sample. So after that we would be going towards find out the particular which particular type of amino acid is present in the sample. So for that we would be performing all these three tests. One is xanthoprotic test, Hopkins test, and then sulfur test for find out the sulfur containing amino acid or indole or aromatic amino acid. After that we would be perform the test acetic acid test to find out the presence of casein in the given sample, and then after that we would be performing the coagulation test. Actually for the coagulation test would give us an idea that the protein that would be present in the sample is actually the albumin. So how we can identify this? They perform this test. For this test we would be having 5 ml of the sample, and we would add 2 drops of concentrated sulfuric acid with the wall, and then we will heat the solution, and after that we will observe and write down the result. And for the confirmation if albumin is present in the sample or not, we would be performing the saturation test, and for this purpose we would be having the 3 ml of the sample, a pinch of diammonium sulfate crystals, and then we will shake it to dissolve, and then we dissolve it until saturation occur. So if this solution gets saturated, so it would be indication of the presence of albumin in the sample. So this is how these are the all tests by which we can finally find out in our sample that in the unknown sample albumin is present. So what we have to do to qualitatively analyze the albumin in the given sample? So first we have to perform the test to find out the presence of to identify the presence of peptide bond in the sample. Then we find out if amino acid is present in the sample or not. Then if we perform the test to find out if the protein is present in the sample or not, and after that when we get confirmation that protein is present in the provided sample, so we would be going towards find out the type of amino acid that is present in that sample in high concentration, and for that we perform these 3 tests, and after that we would be perform a test for the identification of casein in the sample. So of course this test will come negative because the casein is a milk protein. So we have to find out the identification of albumin. So when we perform the other test, that is the coagulation test, so it would come positive, and then we would perform the confirmation test for presence of albumin. So this is how we would be qualitatively analyze the presence of plasma protein, that is albumin, in this sample. So I hope you have understand the how we perform the experiment. So this was about the whole experiment, and thank you very much, and kindly keep watching my videos for the next experiment. Allah Hafiz.